Good evening, everyone, and welcome to this month's event for Atom's live streaming events this year. We've got a really exciting talk lined up for you this evening, and we're really looking forward to welcoming our speaker, and I thank you for joining us tonight. Obviously, things have been changing, they've been developing, and a lot of us look like we might be experiencing a bit more restrictions. So just to reassure you, we are still here and we will still be here for a few months to come, and hopefully next year we'll be able to start seeing you all again in person because we miss you. So just a few things to run through. We will go on to the science snippets in a moment. James will be saying a few words to you this evening before our talk. Specifically, he'll be mentioning about the AGM and a few other things, and then we'll get on with the main event. So if you're ready with your science snippets, everyone, please do start typing them into the comment section, and we will bring them up as they appear. But I will bring in Alphonse and James to join us. There's Alphonse. And there's James. Hi, guys.
Hi there. Okay. So, who's going first?
Alphonse, do you want to go first?
Yes, if I manage to unmute myself, which I think I've now managed. So yes, so if you, just a second. Now you can share my screen. So that's the thing that my wife actually found earlier today and emailed to me, which got me quite excited. So for the first time, people have managed to produce a material that is superconducting at room temperature. So superconducting means it can carry electric current without any resistivity, which is kind of the holy grail of material science. Because if you actually have a superconductor at room temperature, you could just not have all the losses in our power grid and conduct electricity around the world over vast distances. For example, where the sun is, you could get the solar power here without any losses. And what you see here on the picture is actually why this isn't quite there yet and why we have superconducting at room temperature. What you see is the tip of two diamonds, which actually had to compress the material to kind of 150 or 300 gigapascal. So that's kind of 1,000,000 times more pressure than we have here on Earth. And only at those high pressures, you had superconductivity at room temperature. And like the curves you see here on the left, it's actually here as a function of the temperature, which you have at the bottom. It shows you what the resistivity is. And here, kind of, this is on the right-hand side where the number 267 is. That's where the material, if you cooled it down, went to zero resistivity. So that is the material that holds the record there. But again, this 267, that's the pressure, that's the number of gigapascals you needed to make it work. So wow. Quite exciting, but not quite there yet for an everyday application.
Very cool. James, I know you had a few.
Yes, well, that's very exciting, Arthur. I'm thrilled by it. Those of you that have heard me give snippets before or seen me will know that mine are always slightly quirky, and I've got a couple for you tonight, both of which begin with a question for you, really. The first one is, do you ever use strong language when you're driving your car and another vehicle cuts in front of you or brakes sharply in front of you, causing you to brake sharply? And if you do that and your passenger then asks you to calm down or cool it, and you somehow find that difficult to do, you may feel a bit guilty. But scientists at the University of Valencia in Spain have analyzed this and have found that it isn't your fault at all. It's in your genes. You are predisposed to use strong language in those circumstances. So the next time you find yourself in that situation and your partner or your passenger tells you to cool it down, you can tell them, it's not my fault, and carry on yelling at the other car as loud as you like. So I thought you'd be interested in that one. The other one is, and again, begins with a question. I found this one really quite intriguing. Have you ever carried a mug of coffee across a room or anywhere and not spilled any. I haven't, and I bet most people have had that experience of spilling a drop over the side of a mug. On the other hand, if you've carried a glass of wine across a room or a bar, you probably haven't spilled a single drop. So why the difference? On the other hand, if you've carried a glass... Well, scientists in the University of Seoul in South Korea have found the answer. It is to do with the harmonic oscillations of the liquid. And they have analyzed it using what they call Euler-Lagrangian equations. And the paper, and I've glimpsed at it, is 73 pages long. It's been submitted, and it is full of the most obscure mathematics. And I would thoroughly recommend it. It's a lovely bedtime read. So those are my two snippets. Thanks very much. Okay, back to you.
Thanks, James. We've had one come in from Bob Anderson, so I've pulled that up on the screen. So Bob Anderson said, before his 13th birthday, Jackson Oswalt fused together 2 deuterium atoms using a fuser he had built in the playroom of his family home in Memphis, Tennessee. Jackson officially became the world's youngest person to achieve nuclear fusion and will appear in next year's Guinness World Records edition. That is fantastic. I'm definitely not doing that kind of stuff when I was his age. That's amazing. That's really cool. So I'm not sure. I don't think we have any more coming through. So what we might do then is we'll move swiftly on to James, if you want to deliver your messages to our members.
Okay, well, thanks very much. I'll do that. Well, first of all, good evening, everybody. I hope you're all well. It's nice to see you again, even if it's virtually. Well, it's almost AGM time again, and our AGM this year is in November, on November the 19th. And what I'm going to be saying tonight will be part of what is in my chairman's report, which will be circulated to you by Secretary Danielle well before the AGM. The AGM this year will be conducted in a combination of emails and online because it's clearly going to be impractical to have our normal meeting in a hall. So on the 19th is our AGM, but apart from the AGM, on the same day, we have a fascinating talk by Barry Kellett, who is a space scientist at the Rutherford Appleton Laboratory, and he's going to be talking about how over the years, science has been influenced by art. Now, that's going to be a fascinating talk. Now, but before we get to tonight's talk, then there's something I need to say. I have decided, after some thought, to step down from the chairmanship at the AGM this year. The reason is really very simple. I founded this society in 2014 along with the Atom Festival. And for some years I was chairman of both, but I stepped down as chairman of the festival at the beginning of last year, but kept the chairmanship of the society. Well, as you can tell from my picture, I guess, I'm not getting any younger. And I felt that in any case, it was time for a change. So although I am relinquishing the chair, I am hoping to remain on the committee and give my full support to the next chair. I just want to say that I have thoroughly enjoyed my time as chairman, and I am hugely proud of what we have achieved as a society. It's been a great pleasure to meet so many people who are interested in science and technology, and who have hosted so many wonderful and enlightening talks. And as a consequence, I am very grateful indeed to all our speakers, and I owe them all a huge thank you. Throughout my chairmanship, I have been backed by a committed, a loyal and very hard-working committee, and I am extremely grateful to them for their unswerving support. Now, this is the important bit. I shall be nominating Dr. Danielle Fairbrass, our secretary, to be our next chairman, although I guess we should use the term chair. And I hope that all members will give her their support. The entire committee endorsed this nomination, and I do hope that members will endorse it too. Danielle has been an outstanding secretary, and she has hosted our streamed talks, including, of course, tonight. She is young, she's full of energy and ideas, and unlike me, she is a proper scientist. I am quite confident that she will make an excellent chair. With the exception of Victoria, who has managed our publicity so brilliantly in the last couple of years or so, and who is now leaving to take up other interests, I am pleased to say that all current members of the committee are willing to stand again for reselection, re-election, and we would certainly welcome some additional members, and we would dearly love to hear from any member who would like to take on the role of secretary, because that's going to be vacant, and that of publicity officer. Anybody who comes forward to offer themselves for either of those positions will have our undying gratitude and will receive a free atom pen. So anybody who would like to join the society that hasn't yet joined, watching tonight maybe, then go to the website and you'll find there how you can become a member of the society. We welcome you. And finally, it just remains for me to thank all of you, members and supporters alike, for your continued support for me personally and for the society. I shall, as I have said, remain an active member, and I'll be attending all the public talks once they resume, and I hope that can't be too long away, and of course the stream talks in the meantime. And I look forward to seeing you all again whenever that becomes possible. So now, Danielle is going to be introducing Nick Page, our speaker tonight, so I'm now going to hand over to you. Enjoy the talk. Good night.
Thank you very much, James. Thank you, of course, for your kind words and your nomination, and I look forward to the AGM, and we will see what members think. Okay, so on to the main event. Joining us tonight, we have Nick Page, who is the Chief Operations Officer at Oxford Biomedica. Nick comes to us with more than 35 years of experience in manufacturing and supply chain operations and management in highly regulated industries. After gaining his MBA from the Open University in 2001, he has worked for such big names as GE Healthcare and Novartis, before joining OXB about a month before I did in April 2018. And he will be talking to us tonight about the challenges in viral vector manufacturing, working to fast-track vaccine vector production. So welcome, Nick. It's lovely to have you here, and the floor is yours.
Thanks, Danielle. Can you hear me? Yeah, good. Thanks, Danielle. Just by way of a quick introduction to myself, I'll be totally out of my depth scientifically tonight. So if there's any focused scientific-based questions, I'm sure I'll be reaching out for asking Danielle to answer them for me. I just make things. That's what I've been doing for 42 years now, since I started working in a variety of industries, starting off API manufacturing through sterile injectables, aseptics, oral solids, modified release, radiopharmaceuticals, antibiotics, and now viral vectors. So a lot of experience, been around, time in China, time in India, time in Germany for various organizations, and a couple of global roles along the way. During which time I was involved in one startup of a greenfield site, a $500 million investment in India, and also remediated a site in the UK that was under an FDA warning letter. So I spent a lot of years living on adrenaline, and the current situation here is no different. If I may indulge, if I can scroll down, I will just quickly have a few words of introduction. Hopefully most people have heard of Oxford Biomedica, but I'll say a few words about us anyway, just to remind you of what we do and what we're involved in. In addition to our vector supply for Kymriah and other Novartis development programs, we partner with multiple other organizations for lentiviral vector development and supply, most recently with the adenoviral vector supply for AstraZeneca's AZD1222, which is undergoing its clinical trials, and we all hope for everyone's sanity, I think, that it's going to be approved soon. We've got over 600 employees, of which some 250 are involved in product and platform research and development. We've recently expanded our laboratories and our manufacturing facilities, and the pending development of a new innovation center. And we're ideally positioned to support vector programs and scale up from lab initial research through to commercial supply. As a company, I'm just on the wrong screen. As a company, we've been around for 24 years now, just about, focusing on everything viral vector related. We've got a very strong world-leading lentiviral vector platform built up over many years of IP, patents, know-how, and expertise, strong quality systems. We're MHRA and FDA approved across our facilities. And based on that platform technology, we are developing a number of products, gene therapeutic products, in our own pipeline, and some we have out-licensed to other organizations to develop further through the clinical trial program. We also leverage our platform process and understanding to support other manufacturing partners programs, such as Novartis, Sanofi, AstraZeneca, BMS, Juno. And so we've got a really nice business model because we've got the basic platform, we're innovating all the time for our own products and our own processes, and we are learning through working with our partners, which helps us build the platform and develop it stronger for the future. We're building our know-how in multiple therapeutic areas, including gene-modified cell therapies, such as CAR-T, where we're using the vectors to modify cells ex vivo, and also several in vivo applications for our lentiviral vectors, including in the eye, in the brain, in the liver, and with a partnership looking at cystic fibrosis in the lungs. So an awful lot of very clever work going on with the 250 plus people in our research organization. We're innovating everywhere across our business, from cells and vector development, through packaging producer cell lines, through yield improving and process enabling technologies, through analytics, through understanding better how to run the processing, all the way through to artificial intelligence and machine learning with a partnership we've got running with Microsoft. So built on all of that, we've got an awful lot of knowledge and capabilities across the viral vector landscape. And quickly looking at our facilities, we have several laboratories and manufacturing facilities currently approved in Windrush Court, Harrow House, Chancery Gate, and Yarnton, all around Oxford. We're at the planning stage for our big investment in a new state-of-the-art Windrush Innovation Centre, which we should start work on next year. And we've recently opened and got our MHRA licenses and approvals for 4 viral vector processing suites within our new facility called Oxbox, which is the old Oxford sorting office, which we took over a couple of years ago and have just fitted out and got our first licenses. So currently about 226,000 square feet of laboratory and clean room suites. So I was asked, could I talk about challenges in viral vector manufacturing, and then have a little bit of an additional topic around trying to support the vaccine work, which is ongoing. So very quickly, I'm sure you all know this, but I'll remind you anyway. Characteristics of viral vectors. It's a method for delivering DNA and incorporating DNA into target cells. And with lentiviral vectors, particularly good at doing that into non-dividing cells. So our vectors are generated from living biological processes, which make them very, very sensitive. We have to culture cells, we have to get the right conditions, we have to maintain them, and then we have to generate the viral particles, separate them, purify them, and store them in a way that they will not degenerate until they're used. The viral particles, particularly lentiviral particles, are very sensitive themselves. They're sensitive to heat, to shock, some to light. So they require very, very gentle processing and low temperature storage and distribution. Typically, most of the products and the vectors we'll be producing will be stored and distributed at -80. They're inherently difficult to sterilize. You cannot use heat or other strong methods to sterilize the products we're making. And sterile filtration at 0.2 micron is possible, but it's challenging itself because the particles typically 100 to 150 microns in size. And as these are going to be used in vivo or ex vivo, which becomes in vivo applications, they do need to be sterile or produced totally aseptically if we're not able to sterile filter them. The process itself takes a long time. I'll show a schematic of what the typical processes are in a minute, but it takes several weeks from thawing the cell, so the vial of cells from the working cell bank, through growing, culturing, passaging into bigger and bigger flasks and then bioreactors, and then transfecting them to get the viral vector production, and then purifying, separating, and concentrating the vector particles. So that is a several-week process. And while we're doing those several weeks, in any one of our clean room suites that are occupied, we can only have one product. So it makes it very, very facility and people intensive. Once you have made it, the QC testing process, again, it takes a long time. Some of these methods do take days. In fact, some of them take weeks in terms of passaging cells and getting to a position where you can actually get the results. So long production time followed by a long testing time. And one of the other challenges we face, it's a relatively new technology. It's not a big scale technology. So a lot of the materials and component supplies do not have very big commoditized supply chains. There are a few people supplying them, and some of the materials have not been custom designed to actually produce lentiviral processing. They're more standard bioprocessing, and we've had to adapt and compromise with some of them to make them to work in the environment we need to work in. That's improving as more and more programs go through the clinic and start to become commercial, but still we kind of ride on the shoulders of the monoclonal antibody or the other bioprocessing parts of the industry in terms of getting components and materials. Looking at, and I'll show two typical types of processing. The original and more traditional processing is adherent manufacturing process, where you are growing cells up initially in vials and then flasks and then in cell factories. These are cells which have to grow attached to the cell factories. So you gradually grow them, put them into bigger and bigger containers or larger and larger cell factories through several passages. All the time you're feeding them, you're giving them the right gas, the right environment, the right pH, the right conditions, and hopefully you have happy cells which continue to grow, and then you can transfect them, induce the vector production, and then after, in this case, 2 weeks, there's an awful lot of activity then crammed into the next 2 days. So 2 weeks growing the cells up, and then 2 days harvesting the viral vector that's produced, clarifying it, removing residual DNA and unwanted DNA, filtering, and then going through the purification, the concentration, and the final formulation, and then the controlled rate freezing to get the bulk product ready, which will then be sent off to a fill finish process where it will be thawed, sterile filtered, potentially concentrated or reformulated again, ready to be used, and then filled into vials or bags, and then frozen again, ready for testing prior to distribution and shipping. So if you can imagine the picture here, you've got a few cell factories, you run the process for a few weeks, and at the end of it, you get a few liters which has vector particles in there, which depending on the treatment might be enough for somewhere between 2 or 3 to maybe 50 patient doses, depending on if it's an in vivo application needing a lot of particles, or an ex vivo CAR T one where perhaps you need less particles to infect the cells. So a long process, labor intensive process, but not a particularly large output. The next generation of process is a significant improvement, and this is our more standard process we'd be running. This is where we've got a cell bank which can grow in suspension. So here we can grow up in vials, shake flasks, and then through a series of ever bigger bioreactors to get a much, much bigger, more productive process. But you can see here we're growing these cells for 3 weeks from a vial to the bioreactor until we then go through the transfection and the isolation, separation, and purification process. So in this case, nearly a 4-week process. And during that time, you've occupied the clean room suite with one product, at the end of which time you can VHP the whole suite, check it's clean, check it's sterile and ready to go, and then you can put the next program, next batch in. So it's labor intensive, facility intensive, but this particular process has the advantage over the adherent in that it runs without any animal derived serum. So it's fully chemically qualified. It doesn't have any animal products going in, and it delivers around about 10 times the output from the equivalent time than the old adherent process. And there's a lot of work being put in the development and research areas to make this suspension manufacturing process even more productive in terms of either packaging or producer cell lines to get multiple harvests for a period of time to increase the output. And that is very, very key going forwards as we look at trying to drive the affordability and bring down the cost of treatment of some of these viral vectors and gene therapies. I have pinched the next few slides from a presentation I gave at a conference, which really was around what does it take to go from the lab and early clinical studies to commercial scale manufacture. And I pinched a quote from Winston Churchill, blood, toil, tears and sweat. I don't think our manufacturing manager would disagree with that. But what it really takes is a very sound technical scientific knowledge and capabilities, a very solid understanding of the process and of GMP requirements, collaborative team working across all functions, a compliance mindset, which is different from some of the mindsets that happen in certain research organizations, I know, because it's very, very controlled, very focused, very rigorous, and a sizable investment in facilities, infrastructure, validation, equipment, and the team to actually run it. And if anyone follows gene therapy or bioprocessing announcements, you will notice in the news there are many, many facilities announced that they are being built. But from my experience, building a facility is very easy. You take your checkbook out, you find a contractor, you write them a very large check with at least 7 zeros on it, and they hand you over a very nice looking facility. But making it work is what really adds the value, and is actually the most complicated piece of the challenge, is actually getting all the equipment to work regularly, people trained in such a way that the process delivers day in, day out in the same way, in a way that satisfies the regulators. We do hear, or if you look online, you will hear of a lot of new products being delayed because of CMC, so chemistry and manufacturing control issues. This is where you might have a very, very good product that has been developed. It's ready to be launched. The application is with the agency, but the agency hold on to it without approving it because they are not happy about the state or the reliability of the manufacturing facility. So if you look at, again, an FDA website, you'll see lots of compliance actions where there might be a perfectly good product, but the facility, the GMP compliance, the culture of compliance is not there to actually get approval. One other challenge we've seen getting to commercial supply is many of the gene therapy products are given fast track status by the regulators. And because of their relatively low volumes and low number of batches, there are very, very few chances to practice to get the process right as part of that process. So accelerated timelines pose a challenge because everything is done in parallel rather than in sequence, which is the traditional pharmaceutical way. And I'll come on a little bit more on that when I talk about some of the work that's ongoing on vaccines. So what we find is commercial scale manufacturers being established with very limited data, very limited experience on number of batches, which means that it's not perhaps as robust as it could be. The processes we're using are new, not just for the industry, but also for the regulators. So when we get inspections, effectively our sites and our facilities are being used as training courses for GMP inspectors, because most of them will not have seen a viral vector manufacture in anger. Also poses us challenges in getting the regulators to understand the process we're actually running. So looking at a bit more detail, some of the things some of the challenges we face with development capabilities. We see, and not just viral vectors, but a lot of pharmaceutical manufacturing processes, something is developed in the lab which works to an acceptable standard for clinical scale or for research purposes, but isn't robust, isn't necessarily as repeatable as it should be for real, robust, approvable, larger scale processes. So having that real detailed understanding of what it takes to make a process robust is key. Again, some clinical stage programs in other countries don't always need sterile filtration or sterility assurance in the same way. Going for full-blown production, particularly in the UK or a European approved facility, needs sterile filtration and the validation of it. The productivity of viral vectors is a challenge. The adherent process I mentioned, you can have hundreds and hundreds of small-scale cell factories in a facility, very, very labor intensive, very difficult to control to get a significant scale. That's what we call scaling out, just bring more and more of the same small-scale process, or you can develop, as we have done, a suspension process and eventually a producer cell line process, which gives you much more productivity. Might take longer to develop, probably costs more in terms of equipment and systems investment, but will give you bigger and more productive batches. And the process yields, because it's an emerging technology, some of the purification methods used, some of the processing methods used have not been fully optimized. So there is a long way to go to keep on optimizing and get better yields to improve the efficiency and the cost efficiency of the products. Another challenge you always face in manufacturing is when people are developing small scale in the labs, they will use anything they can get their hands on to make it with. Quite often, that will come from small suppliers who cannot supply in large scale or who don't have full-blown GMP facilities, equipment, or capabilities. So getting a real robust supply chain for materials and components is really quite difficult, particularly in an industry like ours, where it has not been around very long and there are not many alternatives in terms of some of the supplies. The process economics needs to be viable for viral vectors. More and more regulators are looking at the pharmacoeconomics of a treatment or of a therapy, and viral vectors are not cheap. I think anyone that's looked at the news will see that Novartis's Kymriah, I think it's, depending on which country you're in, it's of the order of $300,000 a dose. That sounds fairly mind-blowing, but when the alternative treatments probably cost more than that, and all they are doing is prolonging life for a little bit, and the patient is going to end up dying, then $300,000 actually can be seen to be quite an economic cost. But the whole industry is working on trying to reduce that cost to make these treatments much more readily available to the patients. Anyone that's been involved in GMP manufacturing knows that process and analytical characterization, validation, and computer systems validation really are incredibly labor intensive and incredibly focused on by the regulators. It's not just a case of buying a bit of equipment, plugging it in, switching it off, and making your product. You have to prove that everything about that equipment is robust, reliable, repeatable, uncorruptible, that the computer control systems can't be bypassed, that you've got the right logs, the right administrative controls, everything is recorded, and it will always perform perfectly. Proving all of that takes an awful lot of time and effort to get right and to get into a way that the regulators will accept. Understanding what are all the critical process parameters, normal operation ranges, and acceptable ranges of a process is a big piece of work, particularly on something that may have been really rushed through the development process to get it to manufacturing at scale. And then again, developing robust technical transfer processes from laboratory scale through development scale up into full-blown manufacturing, again, is a long and complex process. And what we have found, and we're dealt with it pretty well, but the scale of equipment actually available for some of the viral vector processing has not really been developed. And the range and the capacity for some of the equipment providers and component providers to actually supply bioreactor bags, filters, cartridges at the speed and in the numbers that are required is a little bit of a bottleneck. Again, because this is a fairly new technology and they're not really set up to produce at scale. In terms of plant, a typical GMP facility from concept through implementation, startup, build, equipment qualification and regulatory approval. I think the fastest I've seen it done is about a year. More typically is 3 years. So trying to understand what an organization's requirement is in terms of volume 3 years into the future on products that are probably in development and clinical trials and not yet approved, which may or may not come through to fruition, is quite an art. So trying to have the crystal ball, build new facilities 3 years ahead of when you think they're going to be needed is it's a little bit like going to Vegas and putting your money on red. You've got to make the choice. What do you think is going to be there, and then believe in it. Again, laboratory automation. A lot of the testing we do is very manual. Involves repeat pipetting. Involves many manual manipulations. And so as an organization, we're developing our own automation systems which can cope with improved scale, improved efficiency, improved precision, and also reduction in manual handling and potential repetitive strain issues for the people working in the labs. As an organization grows, you also need to look at the computer systems and the digitization of the business. When you start off, paper and Excel spreadsheets is fine. When you're suddenly up to 600-700 people, multiple processes, multiple customers, and thousands and thousands and thousands, millions of data points, you need to look at digitizing, integrating systems, computer-controlled equipment, and electronic documents, or you get buried in a sea of paperwork and checking. Looking at continuity of supply, for our customers and for patients, you want to have more than one source of supply from your supply chain, but also from your actual plant that you're producing the product in. Things do go wrong in manufacturing. After 40 years, I have seen a few fires in facilities. I haven't started any, but I've seen a few. They do happen. Earthquakes happen. Tsunamis happen. And I've been affected by all of those in my career in one way, shape or form. So trying to establish multiple supply sites for continuity of supply and business continuity purposes is fairly key. Putting in place storage, and it's not just a simple warehouse with racks. It's capable of running to GMP standards. You've got cold store, you've got controlled temperature store, and you've got cryo stores, all the way down to minus 150 for some of our cell banks. So you've got a full range of storage conditions, all of which need to be maintained, all of which need backups, all of which need maintenance, and always looking 3 years ahead about what is the capacity going to be, so you don't run out. So getting the plant right, planning early enough, investing in digitization, and making sure you've got continuity of supply, absolutely key. Other challenges in a fairly new advanced manufacturing area is getting the right people in place. We've been growing very rapidly in an organization. When I joined 2.5, nearly 3 years ago, I think we were 240 people. We're now up, and going into next year, we'll be close to 700 people in the organization. So we've had to go out to find whatever experience we can, but if you look for viral vector manufacturing experience, there is very little out there. So then you have to get the people in and train them in what you need them to understand on the job, because it's very difficult to buy in some of those skills. Recruitment and training timelines are also can be quite excessive. In a GMP environment, in a clean room operation for aseptics, typically anywhere between 3 and 6 months training and validation and media verification you need before you can have someone actually running the process. And if you add on another 2 or 3 months notice period, if you're recruiting someone from another organization, potentially it's nearly a year from starting the process to when you have a fully fledged trained and validated operator. And in terms of knowledge for all the other little bits and pieces, support for regulatory questions, scale up knowledge, and ability to convince auditors and regulators when they come in that you are fit for purpose takes a lot of focus, particularly when you're going through your approvals for the first time in a new facility. I've mentioned a little bit about supply chain, but in our industry, it is a big challenge and focus area. We're not just moving simple things around. We're moving licensed GMP products. We're moving cryogenic stored products. We're shipping in dry ice. We're going across borders. We're going around the world. We'll probably have a few extra complexities and delays tied up with Brexit going to hit us. We have to make sure all of our suppliers are audited, are approved, are performing to GMP, and we have the right contracts in place. We spend a lot of time on business continuity planning, the what-if scenarios, trying to get dual sourcing where we can, trying to make sure we've got enough safety stock in case of interruptions, particularly relevant around the pandemic that we went into, given there's a lot of personal, even basic things like PPE suddenly came in very short supply because the whole world was trying to buy them from a China that had shut its doors to manufacturing at the time. Anything that's being shipped around product related in the cold chain needs the transport system and process to be validated. So we have to do temperature studies using dummy shipments, simulating transport, simulating lorry journeys, flying things around the world just to prove we've got a robust system. Having enough space to hold our safety stocks, having enough supplier capacity in what is a very fast growing industry. And I'll mention a little bit of that when we come on to vaccine as well in a couple of slides. And do we understand what our suppliers, what the second tier of suppliers risks are? Because I've been in organizations where you have what you think are 2 suppliers, but they both obtain a critical material from one other supplier that happens to be in an interesting part of China where the government decide to shut them down for environmental reasons. So really understanding that deeper supply chain is key to maintaining the continuity. I've mentioned cryogenic shipments, but planning, validating these is key. Temperature monitoring and tracking. Shipments will have temperature loggers in there, so you are recording it. You might have minor excursions to deal with to justify. The processing we're actually performing, because the product is very temperature sensitive, our processing steps in the clean rooms have time limitations. Typically, you can have the product at a certain temperature for a few hours before you must have got it through the next stage and got it into the freezer. Very difficult if there are any issues to comply with those limitations to avoid yield loss. Making sure there's enough capacity, backups, etc. on freezers. Again, potentially with the vaccine coming in, we've suddenly had to probably add 10 times our freezer capacity than we had in the organization in very short space of time. When you're dealing with sensitive products, every time you freeze and thaw them, you run the risk of damage if it's not done in the right way. So you have to be very careful about controlled rate freezing and thawing. And it's always a risk in terms of speed versus yield. Customs delays, moving things in and out of countries around the world when they're temperature controlled storage, potentially very easy for the dry ice to run out, the handlers to forget to top it up, and you can lose a whole batch worth of product. Brings us on to the question of when you have a batch of product that could be worth north of £1 million. Do you put it in one shipment? Do you put it in one box? Do you spread it in 6 boxes in 6 different lorries just in case? All of these things kind of have to be taken into account. And then maintaining liquid nitrogen, dry ice supplies, and the handling thereof is also a challenge. When you're using a lot of nitrogen and dry ice in your facilities, you have to have additional oxygen monitoring, people wearing oxygen monitors, lone worker alarms, and everything else like that going on. So again, an awful lot of thought just because of the low temperature handling aspects of the processing we're doing. I don't like scaring anyone, but I mentioned about having a robust supply chain. I was working in one organization when the tsunami hit the Fukushima nuclear facility. And believe it or not, the organization I was with, I think we had 7 sole suppliers within the exclusion zone. So again, something you would never imagine happening. Suddenly 7 of your key suppliers are out of action for more than a year, and you need to find alternatives. The picture below, I'm not disaster prone, but that was from the BBC. That was actually a company I used to work for when one of our facilities, after a contractor had been doing some welding, there was some heat transmitted through a panel which started something smouldering the other side of a wall from where they were working, and during the next few hours it turned into a blaze which actually destroyed the whole factory. And there's a whole host of other things we have to consider when it comes down to a robust chain, a robust supply chain, including serum albumin, which is a limited supply, which is a common item that is used in adherent processing, but there's a limited worldwide supply of. And even when you're using plastic components, some of the big plastic suppliers every now and again will change their process or change their resin. And when you register your product, you have registered which types of plastic is in all the contact parts from all the processing stages. So if any of those change, you have to revalidate the process again using the new tubing, and in some cases get it regulatory approved before that change can be made. And sometimes the bulk plastics manufacturers change their resins without quite enough time to enable you to do that. So I don't want to scare anyone, but supply chain management can be high adrenaline at times. And one in the middle was an interesting one. When I was out in China for 5 years, one of the things that did happen was the Olympics. And during the Olympics in Beijing, factories within a certain mile radius of Beijing were ordered to shut down for about 6 weeks, so the air pollution would go down for the Olympics. No ifs, no buts. If you were in that exclusion zone, you shut your company down. So really good understanding of the supply chain is key. I said I'd mention something about some additional challenges on a fast-track vaccine project. As everybody knows, we're in pandemic conditions, and we were fortunate enough, I guess, to have links with the Jenner Institute, who approached us and a range of other organisations around the potential for producing their ChAdOx vaccine candidate, which has become AstraZeneca's AZD1222 vaccine candidate. Obviously, everybody wants it quickly, and typically the process of scaling up technical transfer, process validation, process proving, and getting regulatory approval could easily take 2 years in standard times. That's now been progressed to about 6 months. And what that means is for the project to start up, normally if you look at a project, you have one critical path. I think this project's probably had 27 critical paths. Everything is on a critical path. The tech transfer activities, the equipment provision, the people provision, the process definition, the analytical definition, the regulatory approval for the facility, everything is on the critical path, and everything is being done in parallel and at risk, because there's no guarantee the candidate will actually get approved at the end. Overlay on that very intense critical path, the pandemic working restrictions, trying to get everything ramped up and obtained, service engineers in to set up new equipment when there are travel restrictions, when you have people quarantining, self-isolating, distancing, remote working, government guidelines. It's been a very, very challenging period for us, during which time, as an organization, we've actually recruited more than 70 staff with remote interviews, with, in some cases, almost remote onboarding, and then gradually getting them in and training them up. But 70 plus staff during the last 6 months hasn't been an easy task. As has been reported in a lot of websites and on the news in various guises, the whole global capacity to produce vaccine candidates of the scale that's necessary isn't really there. And there are supply challenges with everything. With the components, there are multiple vaccines based on viral vector technology or bioprocessing technology, all of which are chasing bioreactors, TFF rigs, filters, cartridges, tubing sets, which to actually make them. So they're almost being rationed. Getting materials during the pandemic period, massive increase in demands for buffers, for processing media, even for oxygen to feed the process. I can remember about 4 months ago when we were struggling to find all the components. We were trying to get in some cylinder banks of oxygen, and we were actually told, sorry, there aren't any. They're all going to the hospitals who are running out. So the challenge is to get even some of the more basic components and gases needed to run the process was a stretch. You have multiple companies all chasing the same suppliers of equipment and components for their vaccine candidates. You have multiple companies contracted to each of the big players in this to try and book enough capacity, contracted to governments, all chasing the same consumables, the same reagents, the same equipment. There is government involvement, and when it comes to vaccines, it would appear the governments are not very sharing. A lot of governments in the world are trying to put their arms around things made in their country and say, we are going to prioritize our country's supply. Kind of understandable, but if you're sourcing components from America, for example, it can make it difficult to get those components shipped to Europe when they're in need for American government supply. Overlay on all of that, we had a facility which we just started up. We had 3 empty clean room suites with no equipment in, and we didn't have regulatory approval for them. So during this process, we've not only had to fit out all those suites, bring in the people to operate them, get the process transferred in and scaled up from small scale into what is now 1,000-litre scale. We've also had to get the MHRA in more than once to actually go through the approval process and audit us and get signed off. And all of these activities have been going on at risk. A massive commitment in terms of cost, a massive commitment in terms of people and resources, and there is no certainty that the trial will be a success. And there's a massive amount of contractual complexity with various equipment providers, government agencies, and the big pharmaceutical companies themselves to negotiate as well. So if I said that the last 6 months have been adrenaline filled, I think it would be an understatement. So we've had the normal challenges of maintaining our ongoing operations, plus 6 or 7 months of adrenaline piled on top. And as I said, 7 months ago, we had 3 empty clean room suites with nothing, no people, no process, no materials, no documents, no license, and no plan. It was unknown what we would be doing. Nobody had even thought of looking at producing vaccine in there. So from 7 months ago with nothing, today we've now got 3 equipped and MHRA licensed clean room suites with process trains capable of manufacturing at 1,000-litre scale. We've added 70 additional staff, well over £5 million worth of additional equipment, some loaned to us from other organisations. such as the VMIC, such as Oxford University, such as AstraZeneca, some bought ourselves, and we hope soon that we will be producing an approved vaccine candidate rather than just a potential one. So the challenges we face to get ready have been many. The challenges just for normal large-scale manufacture, the challenge faced to supply the fast-track vaccine manufacture have been even more. And what I would finish before I throw it open to any questions is just that the staff at OXB have been working absolutely flat out night and day for the last 6 months to try and deliver something that we all hope will help AstraZeneca get the nation out of the sort of the semi-lockdown state that we find ourselves in. And with that, it was a rather non-technical presentation, but I'm happy to open it up to any questions.
Thank you very much, Nick. That was really interesting and really kind of demonstrates exactly how busy everyone on the manufacturing and supply chain side has been in the last 7 months. If anyone does have any questions, please do start popping them in the chat and I will bring them up. But I do have a comment from Alphonse that he has a question, so I will bring him in first. Alphonse.
Yeah, I'm not sure whether it's one or two questions. In the last 7 months, what was your favorite, and maybe that's a different question, maybe not, what was your most difficult problem to solve?
Favorite problem? Oh, there's
No, it's an impossible question, but
I think least favorite has been any negotiations involving any government agency. You may want to delete that from your YouTube recording later on. They're not easy to work with. Most favorite has been working with some of the industry players in terms of the response, the commitment, and the efforts that everyone is going to shift this. It's incredibly impressive what people are trying to do to get this through. I mean, I've been in the industry 40 years, and I don't think I've ever seen things moving at the speed and intensity that this is moving at.
Yeah, definitely. Thank you, Alphonse. I guess I was going to ask a question almost building on that. You touched upon the importance of knowing every single aspect of your supply chain, and even down to knowing where your suppliers are getting their supply from. And I think one thing that became apparent with such things as the PPE, for example, is that it seems that there are certain manufacturers that supply everyone. And one of the key aspects of the vaccine candidate has been that it's been a very collaborative event. Do you think this pandemic will push for more diversity in terms of the number of companies producing things and maybe having lots of small companies producing things rather than big companies, or do you think it won't change?
I think one thing it will drive is governments to take a long, hard look at what their infrastructure is and how much they rely in other parts of the world for their supplies. I know we've seen, and certainly the UK government are interested in developing our own advanced therapies industry going forwards, hence their investment in the cell and gene therapy catapult, hence their investment in VMIC, which is ongoing, to try and have those core capabilities within the country. And with the VMIC to have that core pandemic response capability within the UK. It has also opened up the general question, and it's been opened up previously by other events, but there is not a huge surge capacity available for vial filling in container closure manufacturing. People tend to make what they know they can sell.
Yeah.
And having spare capacity is very, very expensive. So hopefully there'll be a few strategic planners convincing governments that it is worth paying money to reserve capacity just in case, so that the world can respond going forwards. But I think it will drive some long, hard thinking about how reliant organizations might be on one specific country.
Yeah, hopefully.
Yeah.
Okay, I'm just having a see if we've got any more questions coming through. Oh, I think James might have one. Is that you raising your hand, James? Yeah, okay.
You mentioned the supply chain and how international it is, and the cooperation between different manufacturers and companies and research organizations. But what about the collaboration between different political systems in different countries? I mean, one thinks about some of the major international developments like the International Space Station, like CERN in Geneva, where lots of countries have collaborated on major projects. To what degree is the countries of the world collaborating on this? Or is it competitive?
Well, I don't know. I can't really give a very qualified answer, but what you do see is some of the bigger organizations like CEPI across Europe sort of looking at trying to cover European supply rather than individual country supply. You look at some of the bigger, perhaps non-government foundations like the Gates Foundation, things like that, trying to look at investment and support for developing countries. So you've seen some of the more developed countries committing to provide funding for doses, for support for the more underdeveloped countries. But there is, I think a lot of it is every country initially is focused on their own supply, their own contracts getting in as quickly as possible. I think we saw one of the big pharma companies that I won't name, but their CEO got in a bit of hot water because he mentioned that he would prioritize supply to the country he was based in, and then he had to backtrack quite embarrassingly and sort of say, no, we won't do that, we'll provide it to everyone, and try and spread it around a little bit. So I haven't seen a huge amount of international collaboration, but again, I'm not really in a position where I would see that.
I believe there was a WHO initiated agreement that more than 115 countries have signed up to regarding the supply of successful vaccines. But I guess that's, it's not, depends on which country manages to produce a successful one. If not us, then it will be someone else.
And maybe it will depend on who wins the election in America in a few weeks. I don't know.
Well, yes, that's true. Thank you, James. We do have some questions coming from the audience now. So, Jutta asks, will you be able to continue working under an increased infection rate, and will you see an even more difficult situation in supply hunting past January 2021?
I guess the first question really comes down to how COVID secure have we got our workplace, and we put a lot of focus in that. I'm also sort of chairing up our COVID crisis committee and our responses and our actions. So increased infection rate in the community doesn't necessarily transfer to an impact on our organization unless we're not doing the right things within the facility, or unless we have an awful lot of people from outside who are having to self-isolate and quarantine. We are looking at what we do with supply of our own test kits, our own testing capabilities for our workforce, in anticipation that there could be a need to do more testing, more focused, and to try and reduce quarantine and isolation times outside. But obviously we have to follow government guidelines and recommendations where they're in place. We're constantly communicating on people on what are the risks, how to minimize the risks, how to avoid bringing it in, and how to avoid transmission in the workplace. We've had, initially in the pandemic, we had 75% of our workforce remote working, from about 5 people in the company remote working to about 450 overnight in the middle of March, which is quite a strain on IT. But we really are trying to be careful about minimizing multiple site visits, banning visitors, critical people on site only, to try and reduce the potential for any impact or infection transmission amongst our workforce. I mean, looking at supply hunting is actually getting better. We've seen multiple new suppliers coming in. We've seen existing suppliers who can see now the need for these viral vector and bioprocessing facilities to be producing vaccine. We've seen a lot of people investing in capacity, adding people, adding capabilities, adding shifts, adding output. So I think the situation is going to get easier past January, unless there is a total meltdown and every country goes into full lockdown. So it is getting easier. the first few weeks when China shut its doors back in March were probably the scariest, in terms of that's when everyone realized just how little stock was held anywhere in the world and how much of our consumables were coming from Asia.
Yeah. Okay, thank you. I have a question from Jane, who asks, can you use the same cell lines in both the adherent and bioreactor processes?
Well, Danielle, one for you.
I can answer if you wish. Yes, would be the short answer. The adherent processes, the suspension and bioreactor processes use cells that were adapted from cells that were in the adherent processes. Cells that were growing adherently, we adapt them to suspension and then put them to work in the bioreactor process. And the order in which we do that can vary depending on whether on what kind of system we're using, whether it's a transient system or whether we're trying to produce a cell line or a packaging cell line. But the short answer is essentially they're the same cells, but it does require an adaptation of their environment, which can be a lengthy process.
And obviously part of the early sort of cell engineering and some of the work we're doing is around identifying clones, subsets of cells which will perform better than others, so we can continue optimizing the processing.
Precisely. Okay, do we have any more questions coming through? Don't think we have anything at the moment. I'll just ask one more while I wait to see if anyone else has any more. You touched upon the fact that if we're using plastics, we have to have named all the different plastics that we're using, the different resins that make the plastics, and so on and so forth. One question along the line of plastic use, obviously, the environment and our impact upon the environment is at the forefront of everyone's mind at the moment. And I'm sure you know just as well as I do the amount of single-use plastics that go into biopharmaceutical production processes. What do you think our industry can do to reduce our impact on the environment and possibly try and reduce the amount of plasticware that we're using, or do you think that's just not feasible?
And I guess one of the environmental issues we're very, very focused on, energy usage is a big one, water usage is a big one, and the use of plastics is an interesting point, because it's not so much the use of plastics that's the issue, it's the disposal of them that causes all the problems and causes obviously all the things you see on Blue Planet and the mass of plastic in the environment and the microplastics. So what we are very careful about doing is, where we are using single-use plastics, which is fairly standard in the industry, to avoid cross-contamination risks, to ensure you've got a clean product and to ensure that you have got a very, very repeatable system, and to save time, money, effort and resources on cleaning validations, is really making sure that you've got a fully audited, compliant waste disposal route that is not going to put those plastics back into the environment. So typically our plastics will not be going for landfill, they will be going for incineration, probably in a cogeneration facility where it's using the waste heat to try and generate power. So that's our big focus, is where we do have to use plastics, is making sure we are disposing of them in a compliant, audited and environmentally or the most environmentally friendly way we can find.
Yeah. Okay. Thank you very much. I don't think we've got any more questions coming through from the audience. So I guess it's just for me to say thank you very much again, Nick, for your time this evening, and for giving a really quite interesting talk on how we get these fantastic scientific discoveries out into the market. It was really interesting to listen to you this evening. And yep, we're getting thanks coming through on the comments as well. And thank you everyone for watching and joining, and we will see you next time.
Yep, thanks everybody. Thank you, and thanks for inviting me. I enjoyed it.